Bismillahirrahmanirrahim. Assalamualaikum and a very good afternoon to all of you. And welcome to INBIOSIS webinar lecture series 8 in 2022. I am Dr. Nukhalida Kamal and I will be your moderator for today's webinar. I hope the video that we played just now has provided you with some background of INBIOSIS. For those who are interested to collaborate with us, you are most welcome to do. Feel free to browse INBIOSIS websites and social media for the latest updates and info. We feel so grateful today that all of us are granted good health and time to attend our webinar series. So far, this is our 8th webinar for this year. I hope there will be many more webinars to come, and I hope all of you will join again in the future. INBIOSIS webinar series basically was established and started in 2021 for the purpose to provide a knowledge and research experience sharing platform to ensure that we still connected with each other. And we still continue this webinar because of the support that we obtained from our past webinars attendees. So without further ado, I would like to introduce our honorable speaker for today, Associate Professor Dr. Sastia Prama Putri from Osaka University. Allow me to share with you Dr. Sastia's CV. Sastia Putri is an Associate Professor at the Department of Biotechnology, Graduate School of Engineering, Osaka University. Sastia received her Bachelor Science degree in 2004 from Bandung Institute of Technology, Indonesia, while Master of Engineering and PhD degree in 2008 and 2010 from Osaka University, respectively. Her research field is the application of metabolomics for food science and microbiology. She won several awards, for example, L'Oreal Award for Women in Science in 2015 for her work in coffee metabolomics, Osaka University Award in 2018, and Metabolomics Society President's Award in 2020 for her significant contribution to the Metabolomics Society and the fields of metabolomics. Let us welcome Dr. Sastia to share her experience with us. Dr. Sastia, the screen is yours.
Okay, thank you very much. Assalamualaikum warahmatullahi wabarakatuh. Thank you very much, Dr. Nukhalida, for your kind invitation. I'm very happy to finally be here. Actually, you have invited me since last year, and very thankful that you were able to wait until I I gave birth and things settled down a little bit. Yes, so I'm very happy to meet all of you in Malaysia, although not in person. But I hope that I could visit Malaysia sometime again soon, hopefully. And today I'm going to share with you a little bit about what I do in Osaka University. So as have been mentioned by Dr. Nukhalida, that my research is mainly focusing on metabolomics. And today I'm going to talk to you about my research in the application of metabolomics for agricultural products. So I was wondering if you could see my slide already.
Yes.
Okay. So now I would like to start my talk, and if you have any questions or comments during my presentation, feel free to ask me. And we also have a question and answer session right after my talk. So actually, my research is dealing with many different kinds of application areas. Later on, I will talk to you about it. But today, I would like to talk to you something that is probably problematic in both Indonesia, Southeast Asian countries, Malaysia, the Philippines, Thailand, because the Asian countries are very rich in bioresources. We have abundant food production, but I guess we still have a lot of problems regarding crop loss. Originally, I'm from Indonesia, and I thought, how can metabolomics, the research that I'm doing now in Osaka University, contribute to this major problem? So the crop loss mainly is caused by either disease or management, and also and how metabolomics can tackle these issues is we could investigate the cause of disease. So apply metabolomics to like plant pathology, for example. We can also collaborate with agronomists, people from the agriculture science, to aid systematic and effective post-harvest management. And we can work with food researchers or breeding plant breeders to improve the quality of food products. So metabolomics itself, I think many of you who are in attendance probably are already quite familiar with metabolomics. It's a combination of bioscience, analytical chemistry, and informatics. So in order for us to be able to do metabolomics research effectively, of course, we need the knowledge of bioscience, which is the basic pillar of everything. This is where appropriate biological questions will be asked, and therefore knowledge on bioscience is like the fundamental basis on starting metabolomics research. And afterwards, of course, we need a very good command of analytical chemistry, and also organic chemistry, because what we're going to do with metabolomics is to do a comprehensive profiling of metabolites, so these small molecules, which are either byproducts, substrates, intermediates, or products of metabolism. And in order for us to measure these metabolites comprehensively, we need to use, for example, many different kinds of chemical solvents in order to extract the metabolites that are present inside the cell. And then afterwards, we can do the analysis using different kinds of instrumentation. Here, I would be talking to you more about the application of mass spectrometry-based metabolomics. But of course, there's also like NMR-based metabolomics. And we will then generate big data. Once we have big data, it will be quite difficult for us to find the meaning of this big data without the assistance of informatics. So the application of advanced statistics, informatics, is very important for us to easily visualize our data and find meaningful information from this data mining process in order for us to give feedback back to the biomaterials. And then later on, we can do several cycles in order for us to improve a biological system or to understand better about a physiological phenomenon that happens within the bio materials that we are working on. So I would like to emphasize again what is actually metabolomics. It's one of the omics approach that provides a comprehensive analysis of a set of metabolites, which are the chemical component, in which it's directly connected to the phenotype. So we are talking about agricultural products, we're talking about food here. So it's related to taste, flavor, and other food function. And with this connection, we can evaluate the quality and function of food ingredients through what we call metabolite profiling. So Professor Fukusaki, our mentor professor, have already started metabolomics research and applied to many different kinds of Japanese food, for example, like Japanese sake, green tea, different kinds of fermented food like soy sauce, miso, and many other Japanese food. Since the very early conception of metabolomics back then in 2004, 2006. And I started working on metabolomics from 2011. So it was quite early on. We were still a small group, and now we're a group of more than 60 people working on many different kinds of application areas in metabolomics. My research team itself works on the application of metabolomics mainly for unique agricultural products. So for example, like tropical fruits, fermented food like tempeh, perhaps Malaysian also are familiar with tempeh. We also work on animals like shrimp. We work on coffee, chocolate, and nowadays the collection of different kinds of fruits increase as well. Sorry. Okay, so in order for us to do metabolomics, so the process is something like this. We do the evaluation of food quality, and then once we do the evaluation, we also do in parallel metabolite profiling and then make a connection between the two. And then later on, we can identify important metabolites that are related to the phenotype that I understudied. So we can apply this information for manufacturing, storage, distribution, and also, of course, to improve the food sensory or nutritional quality. Sorry. So I would like to show here how metabolite profiling experiment is conducted in our laboratory. So this is Osaka University campus. And this is our mentor professor.
We haven't seen your slides yet.
Oh, the slides is not shown?
Yeah.
Okay. Let me reshare the screen. Okay. So from the beginning, it's not yet seen? Or just?
No, now it's seen. It's
okay.
Yeah.
It was unshared in the middle. Okay. So do you see this slide, the video?
Yes.
Okay, so this is how we do metabolite profiling, and I would like to show you our campus through this short video. So this is our mentor professor, Professor Fukusaki, who established the field of metabolomics. And this is the entrance to our building. So we are under the Department of Biotechnology. This is how our lab looks like. We separate the students' room, study room, and the lab. So each student have their own desk. And this is our lab, where this is the example of application of coffee for coffee beans. So first we do the process of quenching, which is to stop the biological reaction, so stop the metabolism. We do this using liquid nitrogen. In very cold temperature, the metabolism will stop. And then we will grind the coffee beans. Of course, there's like a grinder, but in here we use this multi-bead shocker. And it's important to control this process in order for us to have a homogeneous and the resulting ground coffee beans have to be within the same homogeneous particle size, which will affect later on the efficiency of extraction process. Then we weigh the samples and then extract the metabolites using chemical solvent. In this case, we use methanol, chloroform, and water with a 5:2:2 ratio. Afterwards, we mix it using the thermomixer, certain temperature, and then we will freeze dry and remove the solvent. So this is using the freeze dryer lyophilization process. And then, so still the process of doing the freeze drying. So this is quite important later on, because what we want is just to extract. We would like to remove the solvent while still keeping the metabolites intact. So of course we can do other methods of evaporation, but sometimes it also destroys the thermolabile compounds. Once the freeze drying is completed, we will have this extract remain. You could see there. So here, this is the example of using coffee beans as a sample and for GCMS analysis. So the non-volatile metabolites, we would like to derivatize it first by methoxyamine, and then afterwards we could make it into gas state and apply it for metabolome analysis. So using this derivatization process prior to GCMS, we're also able to measure the non-volatile. So this is one of the examples on the steps on how metabolomics is done. Later on, afterwards, is the data analysis involving multivariate analysis usually. And we work very closely with farmers from Indonesia in order for us to improve the quality of coffee beans. So that's a short video illustrating the whole process of doing a sample preparation for metabolomics. And for the application area that I'm doing in my laboratory is for medical and pharmaceutical, for food, as well as for bioenergy. Actually, even I'm doing metabolomics now. I'm a trained microbiologist. So I work with microbe cells in the beginning as a main research sample. And my first research work as a postdoc is to apply metabolomics technology for microbial strain engineering. And with that, I collaborate with UCLA, with NUS, and also with 𝐓𝐢𝐭𝐥𝐿 University in Germany. And for recent collaboration, we also work, for example, on biopharmaca, which is like herbal medicines in Indonesia. We call it jamu. We apply jamu for quality improvement. We also work with Manchester, University of Manchester, for the research on antibiotics. This is using streptomyces. We work with Harvard University Medical School for the combination between food omics and also epidemiology to understand the effect of dietary intake in a bigger population of humans and improve the quality of food in order to improve the health benefit of food. We also work with a vaccine manufacturer, Biopharma, for the production of biosimilar production for anti-cancer drugs, and many different kinds of collaboration with different institutions, including a chocolate manufacturing company, Pipiltin Cocoa from Indonesia, and a Swiss-based aquaculture company for the shrimp research, Novaton. So I already mentioned the concept of metabolomics is basically to find a correlation between food function and food components. The components itself is measured using many different kinds of tools. In our laboratory, we mainly use mass spectrometry. And for the food function, we employ sensory analysis. It can be using trained panelists or untrained panelists. But what's important is it has to be not subjective. It has to be an objective evaluation and possibly the taste and function and flavors. Usually we employ the sensory analysis and do a combination of this relationship and then find the important metabolites that correlates with the food function. So in our laboratory I have already mentioned, we do a lot of research on different kinds of food. And for the fruit study, we employ metabolomics technology for mango, coconut, pineapple, banana, and mangosteen. So both climateric and non-climateric fruits, we use both. We use it for different characterizations, for example, to study about ripening stages development, and also to understand the effect of a post-harvest strategy like chitosan coating to improve the shelf life of fruit and measure the effect on its metabolite profile, for example. We also care about biodiversity, so we analyze the uniqueness of different fruits coming from different cultivars, different regions, and we also work on this coconut ripening stage and storage condition. For coffee and cacao study, we have a lot of research that have already been published. For example, research on the authentication of coffee, quality assessment, how to find the unique characteristic of coffee and cacao beans coming from different species and geographical origin. Later on, I will be talking a little bit more about this, and to understand the effect of altitude or post-harvest processing or environmental factors and post-harvest factors in metabolite profiles of coffee and cacao beans, and also working with manufacturers to optimize production process. So with shrimp metabolomics study, this is outside of agriculture, but we also work similarly to find objective quality evaluation method and work with aquaculture scientists in order for us to improve the system to cultivate shrimp that is more resistant to disease and also better sensory and nutritional quality. For soybean fermented food, for example, like tempeh and other fermented food, we have a lot of students working on this. So we employ many different kinds of mass spec for tempeh as well. And recently we work on the health benefits of tempeh in the collaboration with Harvard Medical School in order to improve tempeh as a fermented product, as a superfood. We also work on investigating the effect of substituting soybean as a raw material to make tempeh with other legumes, for example, so product diversification, or we make derivatives of the tempeh as a product, for example, as a baby food to prevent stunting in infants, for example. So this is one of the earliest works in coffee. The first work of my food metabolomics project was on the authentication of world's most expensive coffee, which is Kopi Luwak. And this was highlighted in different articles, news articles, as the first method to authenticate the world's most expensive coffee. Then later on, there are several research on Indonesian coffee that I couldn't share with you in detail today, but I would like to show you one example on how we could combine microbiology and also metabolomics in order for us to improve the quality of Arabica coffee beans. So you may know that coffee that is digested by luwak, we already know that there's some digestion happens. There may be some fermentation or microbial consortium inside the luwak gut play a role in altering the metabolite profile after digestion. So what we did was to isolate several bacterial strains from the excrement of luwak, and then we combine many different kinds of optimization. We screen for different kind of enzymatic activities, and then we combine this inoculum and optimize the, for example, time of fermentation, the inoculum ratio to different kinds of coffee beans in order to improve the quality. So here we employ different kinds of fermentation condition to 3 coffee beans coming from 3 different origins. And it's quite interesting to see that the effect of fermentation was able to change the metabolite composition of coffee beans, but it doesn't change it so much that it alters the coffee origin characteristics. So you could see here, for example, the white ones are all Sumedang from Java. They are all clustered together, so that means they have similar metabolite profile. So all of the metabolites, all of the coffee beans that have been subjected to fermentation process are still clustered within its respective control that is not fermented. But if you compare only within one coffee beans, for example, this is Bali, and you compare the control and different kinds of fermentation, you could see clearly that fermentation actually increased particular metabolites. For example, here highlighted is GABA, and many different kinds of precursor metabolites are changed after fermentation. So this is an effective way to modulate the taste of coffee using second fermentation. And in short, we also optimize this process further and was able to improve the specialty coffee that has the cupping score of 81 as a control to a maximum of 85.33, which is a very big leap in specialty coffee. So we proved that this method, combining metabolomics and microbiology, was able to improve the coffee quality through second fermentation. So next, we employ also metabolomics to cacao beans from Indonesia. So Indonesia is one of the biggest producers of cacao beans, but the specialty or fine quality cacao beans are not explored yet from many different places in Indonesia. So I'm interested in this cacao beans because recently in the market we have this bean to bar or single origin chocolates, and it's sold in a higher price, more than 2 times compared to regular chocolate. The difference is because the cacao fruit comes from one origin and the process is called bean to bar. And this is the slide showing the importance of origin. We know that, for example, the same cacao beans, if it's cultivated in 2 different places, it will result in different cacao beans flavor or quality. This is one example that from Krakatoa, we have one from Sumatra, another one from Sulawesi. They have very different taste profile. Even the cacao beans are from the same cultivar. So it's quite interesting to know what are the metabolites that are responsible for these differences in the taste and flavor. So Indonesia is an archipelagic country. It has various geographical features. So we produce cacao beans from Sumatra to Papua, but the research on the diversity of cacao beans from Indonesia is still very, very limited, especially on the fine cacao beans. So our aim is to understand what makes each area unique, and also perhaps we could identify what kind of environmental factors coming from each different origin is responsible for the uniqueness of the metabolite profile of cacao beans from different origin. So as a strategy, the first step is to observe the effect of origins in Indonesia, and the second step is to observe the correlation between environmental factors of origin place and metabolite profile. So this is the workflow of how we do metabolomics, similar with coffee beans. Before we compare different kinds of cacao beans, round it, weigh using the derivatization after extraction, and then employ data analysis. So here we have a very comprehensive analysis of fermented beans collected from major cacao producing area. Here we use 17 origin places, and the farms are the foster farms of our collaborator in Indonesian Coffee and Cacao Research Institute, as well as Pipiltin Cocoa, our collaborator. And actually the fermentation condition is controlled to be same in all regions. So we only select the farms with consistent yield and trained to do cacao cultivation and fermentation. And all of them are during the same harvesting period. What's interesting for us is when we compare different with coffee, in the case of coffee, Sumatra coffee beans are very neatly clustered with each other, whereas the coffee beans, for example, from Sulawesi, from Java, from Papua are clustered differently. With cacao beans, we do not see very clear clustering based on origin. So for example, here, the samples from South Sulawesi and Central Sulawesi are close by, but we have also Central Sulawesi that are clustered together with Bali, for example. So we wonder what makes this basis of sample clustering. So maybe there's another factor. What we know is that there are two major clusters. One is those with higher amino acids, and one with lower amino acid content and maybe higher sugars. And we could see what are unique in different origins. For example, we see here that Aceh cacao beans accumulate highest GABA, which is interesting. Or perhaps Central Sulawesi is high in threonine. It's high in ravinose, and Papua is high, and East Java has high concentration of threonine. So we could then later on develop, for example, if we would like to have cacao beans that are high in this and this, we know where to get or where to source these cacao beans. And next, we would like to observe what is actually the basis of the clustering. So first, we compare the raw bean and fermented bean, and very clearly we could see very beautiful clustering. Raw beans are clustered together while fermented beans are clustered separately. And we could see that the contributing metabolites for these clustering are sugars and amino acids. After fermentation, we see an increase of amino acids and a rapid decrease of sugars, which is very natural because raw cacao beans contain high sucrose, and then maybe these sucrose or glucose, sucrose and other sugars are being used by the microbes for fermentation and generate high concentration of amino acids. So this fermentation has been known to generate flavor precursors such as amino acids, and we could see very clearly here that the fermentation increased amino acids, for example, like glycine, leucine, and threonine, as can be seen in the bar graph here. Oh, I think it's okay, still sharing. Oh, sorry. Okay.
Yeah, we can see it.
Okay, so I think I pressed something and then it's gone. Okay, so within the raw bean, we also see that there's a very interesting clustering of cacao beans that are cultivated in different altitudes. So environmental factors like altitude can probably explain the clustering trend of metabolite profile in raw beans. So we did a OPLS regression model to find a correlation between altitude And we found also it somehow correlates with the temperature, because we know that the higher the altitude, probably the lower the temperature becomes. So actually these two factors are correlated. And we found a very nice model. We could establish a quite nice regression model when we use altitude as a response variate and metabolite profile as the explanatory variate. And we found that higher altitude and lower temperature were correlated with intensity of succinate, GABA, glycine, glycerol, and ethanolamine. So as a conclusion here, we see the diversity of Indonesian cacao beans, and we see that some metabolites are featured and unique to some particular origin, and it might have potential as cacao beans with added value, for example, high GABA cacao beans, natural high GABA. And we also found that altitude affects the concentration of succinate, GABA, glycine, glycerol, and ethanolamine. So very happily, this paper on cacao beans is just recently accepted for publication just 3 days ago. So probably, hopefully, we could share the results with you soon about our first publication on cocoa beans metabolomics. So we hope that these findings will provide a new insight on the specific metabolites that will be useful in chocolate production using Indonesian cacao beans. Okay, so that is a story on coffee and cacao, and I would like to now move to fruits. We did a lot of research about ripening stage of mangosteen, which is a climateric fruits, but we are also now interested in pineapple, which is non-climateric fruits. We have already published a paper that shows us the correlation of inositol and mannose and galactose, in particular from the flesh. Malesitose and inositol are strongly correlated with ripening, in which malesitose increase during the progression of ripening. C0 is unripened and C4 is the most ripe, and inositol decrease during ripening. So we then use this research result and coat the fruits with malesitose and inositol and see that actually by coating it with malesitose, we were able to improve the shelf life of cut fruits in pineapple. I'm sorry I don't show the results here because it's now still under review, but using this kind of information by understanding what kind of metabolites are correlated with ripening and use that metabolites to devise a strategy for post-harvest will result in actually longer shelf life fruits. So this is one of the example on how metabolomics result can be used to improve fruit quality, in this case, shelf life. So we know that, so this is just a background on why that kind of research is quite interesting, is because in Japan, we normally cannot buy C4 fruits because C4 fruits is considered as high-class export niche. The reason is because the C4 fruits, although it's very sweet, it only lasts for 1 week before it deteriorates. And the C1 stage, on the other hand, lasts longer and therefore can be transported using sea freight. But C4 stage lasts shorter time, so it has to be transported using a faster way, air freight, and significantly this increased the cost. So our approach in metabolomics is to first study what happens during ripening from C0 to C4, and then see if we could use some kind of information in order to make the C4 last longer. So this is almost achieved, and it's a research work of one of our doctoral students who will graduate this September. So in short, with the metabolomics, we were able to annotate many different kinds of metabolites, including amino acids, organic acids, saccharides, and other metabolites. We measure the metabolite intensity as well as the color of the peel using spectrophotometer. This is a mobile spectrophotometer. Using the LAB method, we are able to measure quantitatively the color changes. And then we saw that the peel color was changed progressively relative to the C1, C0 stage. And we saw the separation between the ripening stages, both in the flesh. And here we show that all parts of pineapple flesh and peel show clustering and trends based on the ripening stage. And we make a PLS model to make a correlation between the flesh metabolite profile as an exploratory variable, and we use this color changes as an indicator of ripening as a response variable. Then we can find some unique correlation between metabolites and some ripening. And then later we can discuss about why, for example, there's a decrease of amino acids because probably it's converted into aroma compound production towards the end of the ripening. And we also found that these are the competing pathway for ethylene production during ripening. And so we indicate several metabolite importance in the pineapple ripening process. And finally, I would like to show a research result. Also, this one has been published recently by comparing metabolomics between different kind of cultivar. So here we already mentioned that there's a clear difference between C1 and C4 stage. But here we would like to know whether these very different metabolite profile between different ripening stages still occur if we employ many different kinds of pineapple from different cultivars. So here we also correlate the sensory profile of pineapple, and we would like to make a correlation between the component and taste profile to improve the pineapple quality. So here we use pineapple from different ripening stages and different cultivars from different countries. We use the samples from Indonesia and the Philippines, different ripening stages. And these are the RATA test, rate all that apply test for pineapple to do the sensory evaluation of pineapple. We analyze the taste, texture, aroma, and acceptability. And we found, in conclusion, wherever the pineapple is from, or whichever cultivar we use, still C4 pineapple is better. So the most important separation is just between the C1 and C4 fruits, regardless where they are from. So that means ripening stage is very, very important. People will still prefer fruits that are from the C4, right? Because it's sweet and juicy and it has high acceptability according to the RATA test. Then we also found the correlation between the C1 and C4, and we see the diversity between red Spanish and smooth cayenne. And we could see the characteristic metabolites for each ripening stages and cultivar. Okay, please wait. Okay. Okay. Let me continue by showing the prediction model of sweetness. Okay, so we could see what kind of metabolites that are correlated with sweetness. For example, isocitric and malic acid, ascorbic acid, and quinic acid. These are the metabolites contributing to sourness and firmness, whereas the juiciness, sweetness, and acceptability correlates with different kind of metabolites as shown here, some amino acids and sucrose, for sure. So here we could see that sensory evaluation reveal that consumers are more aware on the differences in the ripening stage rather than cultivar, but the metabolite profiling is more sensitive, which enables us to distinguish the differences between cultivar. And we can see that sourness and firmness contribute to C1 fruits, while sweetness, juiciness, and high acceptability contribute to C4 fruits. And we could show what kind of metabolites are correlated with these sensory characteristics. And we also were able to tell the difference between difference in pineapple cultivars by GCMS analysis. So what I would like to emphasize again is that kind of study can provide us a better understanding of the relationship between chemical composition and sensory attributes of different pineapple cultivars and ripening stage. And hopefully this kind of research will be useful for future selection, breeding, and product development of superior pineapple products. So these are the acknowledgement of our partners. And I would like to close my talk by saying that metabolomics is a very powerful tool to discriminate different kind of food products. And we can apply it for example, authentication, seeing the differences between GMO or non-GMO. And a lot of studies also deals with halal science. And we can also do the metabolomics analysis for quality evaluation and understanding, for example, what happens during the progression of ripening, or what kind of metabolite correlates with specific food attributes, as have been mentioned in the pineapple study. And we can also use metabolomics to predict important quantitative phenotypes. For example, we could see what kind of metabolites is correlated with sweetness or juiciness or acceptability. Then once we know what kind of metabolite is important for food quality, for acceptability, then we could target these kind of metabolites and improve it further in terms of its content. And usually it also relates to the increase in its quality. So what's next? I also try to bridge between the research work and apply it to real industry. So myself and my family established a company in Indonesia and work directly with farmers in order for us to apply this kind of metabolomics technology. Here shown is story about coconut. So we were successful to export coconut from Indonesia. And now we are trying to apply metabolomics technology in order to improve the shelf life, storage, and logistic distribution to reduce the cost of transportation so that the cost of goods after export can be cheaper. And also the research on cocoa and coffee was able to give us a more quantitative understanding on the differences between diverse biodiversity in Indonesia. And also this can be applied to understanding the uniqueness of each origin. So this was also resulting in the export of many different kinds of specialty coffee beans and cacao beans from Indonesia from the research that I did. And finally, I would like to thank all of the students that work under this project. Here we have, in fact, one Malaysian student and who's working on tempe. That's Hadi here. And we also have a lot of students coming from other countries such as Indonesia, Thailand, and the Philippines. So I hope some of you who have students or haven't got your PhD are interested to join our laboratory to do exciting metabolomics work. And I would like to close my talk by thanking all of my collaborators in Institute Teknologi Bandung, Indonesian Coffee and Cocoa Research Institute, and Pipiltin Cocoa. So I think that is all for my presentation. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you very much to Dr. Sastia. I still think, 45 minutes is not enough to listen to all your achievement in applying metabolomics, especially in agriculture. So while waiting for the audience to type the question in the chat box, so let me check. We have one question. So I will only read question for 15 minutes. So if we have more questions, then we will forward the question to you later. The first question from Dr. Morni. Great presentation, Dr. Sastia. May I know if we attain the metabolite of each stage from C1 to C4 of the pineapple, how does it work to prolong the shelf life of the pineapple?
Yes, so we work under the assumption that once the metabolites progress during ripening, maybe some metabolites increase or decrease during ripening. So by adding this metabolite, we hope that it will alter somehow the ripening process. For example, some metabolites decrease during ripening. And if we add this kind of metabolite, we thought that maybe it can act like an inhibitor or delay the ripening process um, by adding it to the pineapple. So that is just an idea on how metabolomics can be done. Of course, it's better to it's quite different to work on the idea like endogenous concentration versus addition, but somehow it works when we coat the pineapple with melezitose, which is the metabolite that we found to be increased. It actually improved the shelf life of ripening. So similarly, we found that several metabolites are correlated with the ripening in mangosteen, and we use that particular metabolite and add it to the mangosteen. It also delayed the ripening. So there is somehow a correlation, and we could modulate the process of ripening by adding, because we cannot decrease it, right? So we just add these metabolites that are correlated with ripening. But the exact mechanism, I don't know.
Yeah. That's another study that needs to be done.
Yes.
Yeah, study the functionality of that metabolites, of course. So the next question from Dr. Goh. Thanks for an interesting talk. Metabolomics is a very challenging field with high sensitivity to variations. May I know how do you standardize the samples and what's the greatest challenge you face? Where is it? That you face in your projects?
Yeah, very, very nice question from both Dr. Morni and from Facebook, 𝐓𝐢𝐭𝐥𝐞. So how to standardize the sample is actually very difficult, especially if we talk about agricultural products. Sometimes we even get samples from the market. I have students who work on research and they just um, got the samples from the market. But that is not the point. The point is, regardless of the variation, we still see clustering. That is important. So that means in order for us to non-biasly judge, we actually welcome these variations within our sample and increase the number of sample as much as possible so that when we say it represents Indonesian cacao beans from one particular region, we have to take many, many samples from that region because each region will represent its own variation, right? But even with that variation, we still see the clustering within the same region, then that's fine. That means the classification can still be seen despite these variations. And for us, that is the first step to do metabolomics experiment, for us to generate the hypothesis, see a general clustering. And then once we already have the idea, for example, in the cacao bean study, we found that, okay, despite all of these variations with so many cacao beans, we found that probably the similarity is due to altitude. And this altitude is due to temperature. So now we are controlling to validate this research. We do the next step of experiment. We do the fermentation exactly the same way, exactly the same cacao beans, just in two different locations, having very different altitude and temperature. So this is how to validate the next step. The first is we don't know what kind of variation, and many variations exist. Then once we found the variation, then we can do the validation. So the most challenging is to get a good sample, actually, because sometimes if we don't have a good sample, then we would not know what is the basis of this clustering. Sometimes, for example, like in the cacao bean study, very different with coffee beans, I already mentioned. With coffee beans, very clear clustering, okay, based on Arabica and Robusta, based on green and roasted, based on different origin. With cacao beans, in the beginning, we don't know what is this. And then once we investigate more and more and understand the variations that each region brings, then we could somehow identify these environmental factors. So, but that is a very big challenge. Then the second biggest challenge for me to do metabolomics is what to do with metabolomics study, or what to do with the result. So we know that these samples and these samples are different, right? And nowadays, it's not enough to do just that. If you want to publish in a good paper, it's not enough just to say, okay, A and B are different, and the differences is this and this.
Yeah.
That kind of research probably will be accepted 15 years ago when I started metabolomics
Mm.
research. That kind of study can no longer be accepted in high reputable journal. So after you find the first study result, then that is actually the first generation of hypothesis. What you do with your data, either you improve something or you're able to explain a mechanistic relationship of a phenotype, only then you could publish. So that is actually the most difficult part. So to ask the right biological question and how to interpret the data and use that data to synthesize new ideas that result in a breakthrough, that is, I think, the biggest challenge still for me.
Yep. Thank you, Dr. Sastia, for a very comprehensive answer. Because I think it's quite challenging for us to study metabolomics, especially in relation to environmental factors due to variation. But I think based on your explanation, then we know that first thing, variation is okay. Then we come up from there with what are the relationship between the two, and get more information from our data. So we have another question from 𝐓𝐢𝐭𝐥𝐞. Good presentation. From the presentation, I understand that there is a variation in the metabolites between agriculture produce, fruits and others from region states of it, fermented or non-state, ripe or unripe. My question is, are you in any way considering experimenting the effect of microbes using metabolomic approach to give the insights into their efficacies at different for variation? Yeah, I also want to ask this question.
Microbes effect. Oh yeah,
Yeah.
it's very
Mm-mm.
important. Actually, I did the experiment on uh, the effect of microbe to improve the coffee quality. So that one we isolated some microbe and then we use this controlled fermentation. So we know that it's very important. But do you mean the microbiome in each region? Is that the question?
Mm,
The
uh,
effect of the microbiome?
yes, I think so. Because I think
Yeah.
different, yeah.
Yes. Yeah.
Yes.
Yes.
Mm-mm.
Yeah. I'm interested in that, but it's very difficult.
Yeah, yeah.
Actually,
All right.
very difficult, but we actually have the research on the combination of the microbiome and metabolomics. We collaborate with
Mm-hmm.
NUS as well, and with ITB, but it's quite challenging actually to pinpoint because the microbiome itself is very, very complex, right?
Yes.
So even we find some correlation, it's still difficult to know this metabolite comes from which microbe. That is very difficult.
Mm-mm.
So we can
Mm-mm.
analyze this microbiome is like this as a complexity, and this result in this kind of metabolite profile as a unit. But individual contribution of a particular microbe and a particular metabolite is still, for me, very challenging. There are experts, for example, but it needs very smart and intelligent modeling
Mm-mm.
to find the correlation between the microbiome and metabolome, I think. But this is a very
Okay.
interesting perspective.
All right. We have question from Dr. Lin Changjo. Thank you for your interesting talk. I'm very impressed on your work and especially inspired by your last slide where you have set up a company. How do you manage to integrate research into the real world application? What are the major challenges and how do you overcome them?
Yeah, thank you so much. Actually, I am still
Yeah.
quite new in the research and company business, but I saw that many times throughout the years of doing metabolomics research, and For example, being interviewed by some newspaper and being asked, what is actually your impact to your work? And I realized that my impact is only for academics because, yeah, I publish papers, but then the farmers don't read my papers, right? And the farmers cannot do metabolomics research. So I understand that the gap is so huge. And every time I write a project proposal and write a paper, of course, we have to say this is applicable to industry, can improve something. I feel like it's kind of like a cliche if I actually don't do something. So yeah, finally I decided to set up a company and really talk with the farmers and the people who are actually there to understand what is the problem in the industry, what is the problem in the farmers. So for example, they mentioned that the crop loss usually occurs due to disease, for example, or due to the poor harvest management, or due to the logistics. So by talking a lot with the people who are actually in the business and in the supply chain, starting from the farmers and the traders, the exporters to the consumers, we could understand what is the real life problems that needs answer. And being an engineer, because I'm in the faculty of engineering, so engineers is like service science. So we give service to the community. Basically, our research needs to answer these problems that are in the society. So I think the best way is to talk a lot with people who are in the field. Go there, visit them. I stayed in the people's homes, for example, like small farmers. I went crossing to the forest, for example. There's no bridge and cross rivers without bridges, for example. Yeah, I went to these places and see what are the difficulties, why they have like such a good potential for producing very high quality products, but they cannot. Then understand these challenges and then try to train them and help them in. So I give them incentive. If after training they do well, then they will be priced. They will be. So for farmers, it's very simple. If they have good products, they need to be appreciated. It needs to be purchased in a higher price. Without that, they don't care. They don't want to do more if you're not gonna pay. So I became the person who will value them higher. Basically, that is the key. That is the only incentive that works. It's just financial incentive.
That's the tip.
That is the thing. Yeah, you could say that, okay, be proud of your work, make a good quality product. They don't mind that part as long as it pays and it pays well. That becomes the incentive. Yeah. So I say like, okay, if you want to have a good product, then you need to do a fermentation. You have to do the fermentation this way, and this is the correct way to do fermentation. And I can know if you do the fermentation 3 days or 5 days, I can know from the metabolite profile. Like because I have a marker. If it's full fermentation, this marker metabolite has to increase. So if they don't do that, they cannot lie, for example, because I have the method.
Cannot lie. I tell the truth.
Something like that. Yeah, something like that. Yeah.
Good tips for us. I mean, to in the future, to join. I mean, to maybe be like you, open a company and help the farmer or the small agriculture business holder, sort of like that. Basically, we have several questions more, but it's already 4 o'clock, unfortunately. However, I will email you,